Dear students, let us continue with one more concept in transport in plants. Okay, it is plasmolysis. Okay, and here uh, the in case of plants, okay, or in case of plant cells, the behavior of plant cells. Okay, we can consider the behavior of plant cells or tissues with regard to water movement. With regard to water movement, it depends on the surrounding solution. Okay, the behavior of the plant cells or tissues with regard to water movement depends on the surrounding solution. Okay, and we can mention the different types of solution here. Okay, a solution can be termed as hypotonic, then hypertonic or isotonic depending upon its osmotic concentrations with respect to another solution or cell sap. Okay, we mention the different solutions like this. Okay, hypotonic, then hypertonic as well as the isotonic. Okay, it will be depending on its osmotic concentration with respect to another solution, or we can consider as the cell sap, right? And based on this the different types of solutions are there. Okay, one is what is there, isotonic. Isotonic means what here, if an external solution balances the osmotic pressure. Okay, if an external solution balances the osmotic pressure of cytoplasm, it is called as the isotonic solution, right? Okay, isotonic solution. Okay, and here we can also mention like this. Isotonic solution means what here, a solution whose osmotic concentration okay, a solution whose osmotic concentration is equal to that of another solution or cell sap is called as isotonic solution, right? And here when a cell or tissue is placed in isotonic solution, okay, when a cell or tissue is placed in isotonic solution, there is no net flow of water towards inside or outside. Okay, and such cells are said to be flaccid, right? Okay, when a cell or tissue is placed in isotonic solution, there is no net flow of water towards inside or outside. Okay, it means what here, water flow will be is in equilibrium. Okay, such cells are now said to be flaccid, right? It is nothing but the intermediate state here, right? And one more solution is called as the hypotonic solution. Okay, hypotonic solution means what here, if the external solution is more dilute. Okay, if the external solution is more dilute, okay, it means it will be having higher water potential than the cytoplasm. Okay, and it is said to be hypotonic. Okay, and here when we keep the plant cells in hypotonic solution, okay, the cells will swell, or they show the turgid condition. Cells will swell in hypotonic solution, and for this condition what we call it as the turgid condition. Okay, one we have studied about the flaccid. Okay, then another one will be turgid, right? And uh, turgid condition, or we can also define like this, hypotonic solution. Okay, a solution whose osmotic concentration, a solution whose osmotic concentration is more than that of another solution or cell sap is called as hypertonic solution. Okay, I'll repeat it once again. A solution whose osmotic concentration is more than that of another solution or cell sap is called as hypertonic solution, right? And one more thing is the third type is called as hypertonic solution, right? Hypertonic solution. Okay, what is the definition here? If external solution is more concentrated, if external solution is more concentrated, it means it will be having more solutes than cytoplasm, then it is said to be hypertonic solution. Okay, or one more definition we can mention, right? And here it can also be defined as a solution whose osmotic concentration is more than that of another solution or cell sap is called as hypertonic solution. Okay, I will repeat it once again. A solution whose osmotic concentration is more than that of another solution or cell sap is called as hypertonic solution, right? Okay, and here we are going to concentrate mainly on the plasmolysis process, right? Plasmolysis. And here already we have studied the different types of solutions now, right? And here plasmolysis means what? Here how plasmolysis occurs. Okay, and here when a cell is placed in hypertonic solution, okay, when a cell is placed in a hypertonic solution, it means what? Here high concentrated solution when compared to the cytoplasm or cell sap. Okay, when a cell is placed in hypertonic solution, water moves from the cell, okay, that is from area of higher water potential across the membrane to outside. Okay, that is area of lower water potential. Okay, outside what will be there? Water potential will be low, and inside the water potential will be high, right? And the cell will shrink here. It is called as the plasmolysis, right? And here uh, we can also define like this. Okay, plasmolysis is nothing but the withdrawal of protoplast of a plant cell from its wall due to excessive loss of water from a cell due to osmotic action. Okay, and it occurs when a plant cell is placed in hypertonic solution. Okay, and in very short we can define the plasmolysis like this. Okay, the shrinkage of the protoplasm due to exosmosis. Okay, the shrinkage of protoplasm due to exosmosis is also called as plasmolysis. Okay, and here during the process of plasmolysis, water is first lost from the cytoplasm. Okay, during the process of plasmolysis, water is first lost from the cytoplasm and then from the vacuole. Okay, and another important point is during plasmolysis, the cell membrane and protoplast. Okay, the cell membrane and the protoplast of a plant cell shrinks away from its cell wall, and such cells are said to be plasmolysed cells. Okay, during plasmolysis, the cell membrane and protoplast of a cell shrinks away from its cell wall. Such cells are said to be plasmolysed cells, right? And here in the diagram, okay, in A we can observe plasmolysed cell. Okay, there is a shrinkage of protoplasm. Okay, it will be separated from the cell wall and it will occupy any corner of the cell, right? Okay, and another thing is flaccid cell. The second one is the flaccid cell. It is nothing but the intermediate condition, right? And third one is the turgid cell. Turgid cell because it contains more amount of water. It contains more amount of water, and and here we can perform the experiment of plasmolysis in um, some plants. Okay, say for example in these two diagrams you can observe the plasmolysis process before plasmolysis and plasmolyzed cell, right? And here in a plant called as um, rio leaf. Okay, in case of rio leaf, we can take the epidermal peel and we can perform the experiment regarding plasmolysis. Okay, we can take the epidermis of the rio leaf and we can keep in a small portion of the epidermis on a slide containing a few drops of hypertonic solution, right? And after some time we can observe the plasmolysis process. Okay, otherwise we can take the onion peel. Okay, in case of onion peel also we can study the plasmolysis process. Okay, by keeping the some of the epidermal cells in a high concentrated solution or hypertonic solution, right? And the other thing is let us move on to the next other points. Okay, and plasmolysis is said to be usually reversible. Okay, plasmolysis is usually reversible. It means when the cells are placed in a hypotonic solution, okay, water diffuses into the cell. As a result, the cytoplasm builds up a pressure against the wall. It is called as the turgor pressure. Okay, and here For this process we can also call it as the deplasmolysis. It means it is nothing but the deplasmolysis means what here? It is swelling of a plasmolized cell as to come to its original state. Okay, deplasmolysis occurs when a freshly plasmolized cell. Okay, deplasmolysis occurs when a freshly plasmolized cell is placed in a hypotonic solution or pure water, right? And here this point explains the same concept. Okay, it will explain about the deplasmolysis only. Okay, what they have mentioned here, plasmolysis is usually reversible. When the cells are placed in a hypotonic solution, water diffuses into the cell. As a result, the cytoplasm uh, builds up a pressure against the wall. Okay, now the pressure which is developed in from the cytoplasm here, okay, it will develop against the cell wall. It is called as the turgor pressure, right? And here if the turgor pressure develops in the cell, there is no any breaking of the cell here. Why? Because the cell wall will be rigid here, right? And try to understand the next point. Okay, the pressure exerted by the protoplast due to entry of water against the rigid cell walls is called as pressure potential. Now this is also called as the pressure potential, and it is mentioned as psi p. Psi p. Okay, and what is the third point here? uh, the cell does not rupture. The cell does not rupture due to the rigidity of the cell wall. Okay, the cell wall will be rigid and hence it will not rupture, right? And turgor pressure causes, okay, what is the importance of turgor pressure here? Okay, if the turgor pressure develops inside the cell, it causes enlargement and extension growth of the cells. Okay, it is responsible for the cell enlargement and growth or extension growth of the cells. Okay, this is the importance of the turgor pressure, right? Okay, and this is regarding the concept of plasmolysis. Okay, and finally in textbook they will ask you one question, right? What is that question? Okay, during this concept, right? And what occupies the space between the cell wall? What occupies the space between the cell wall and the shrunken protoplast in the plasmolyzed cell? This is one question, right? And here the answer for this one is the external hypertonic solution. Okay, the external hypertonic solution occupies the space between the cell wall and the shrunken protoplast in the plasmolyzed cell. Okay, this is the correct answer, right? Okay. Then next one. Okay, one more question is there in textbook. What would be the pressure potential of a flaccid cell? What would be the pressure potential of a flaccid cell? Okay, and here the answer will be like this. The value of pressure potential, okay, it would be more positive for a turgid cell. Okay, the value of pressure potential would be more positive for a turgid cell, and while it will be zero or very negligible. Okay, it will be zero or negligible for a flaccid cell. Okay, pressure potential for the flaccid cell will be zero or negligible, right? Okay, this is regarding the plasmolysis, right? Okay. And one more concept is there. Okay, it is nothing but the imbibition. Okay, what is imbibition here? It is a type of diffusion. Okay, or we can mention it as a special type of diffusion when in which water is absorbed by the solids or colloids, increasing their volume. Okay, it is a type of diffusion in which water is absorbed by solids or colloids, increasing their volume. Okay, this is called as the imbibition, right? And here example, we can mention one example here: absorption of water by seeds and dry wood. Okay, we can mention this one example. What is that one? absorption of water by seeds and dry wood. Okay, and one more example we can give here. The pressure due to swelling of wood can split rocks. Okay, and in the ancient times, the people used to split the rocks by using this method. Okay, they used to in the crevices or in the cracks, they used to put the wooden uh, wooden sticks. Okay, and they go on adding water to them, and because of the imbibitional pressure developed by that wood, it can split the rocks. Okay, that pressure is responsible for the breaking rocks here. It is called as the imbibition pressure. Okay, the pressure due to swelling of wood can split the rocks. Okay, this method was used by prehistoric man to split rocks and boulders, right? And here, the one more example. Seedlings are emerged out of the soil due to imbibition pressure. One more example. Okay, the seeds will germinate. Okay, at this time they will emerge out. The seedlings will emerge out of the soil. It is mainly due to the development or creation of imbibitional pressure only. Imbibitional pressure, right? And for imbibition, we can give some other example. Say for example, the swelling of the wooden doors and or wooden frameworks during the rainy season, right? Okay, like windows and doors. Okay, it is also mainly due to imbibition only, right? And here uh, one more point. uh, the conditions, okay, necessary for imbibition. Okay, what are the important conditions for the imbibition to occur, or the requirements? We can mention them as the requirements. Okay, imbibition requires. Okay, imbibition requires what here? Difference in concentration gradient. Okay, difference in concentration gradient. Okay, it means what here? Difference in concentration gradient. Okay, it means the water potential gradient between the water potential gradient between the surface of the absorbent and the liquid imbibed. Okay, and the liquid imbibed. Okay, water potential gradient between the absorbent and the liquid imbibed. Another important requirement is affinity between the absorbent and liquid. Affinity between the absorbent and liquid. Okay, and here with regard to imbibition, some important terminologies are there. Okay, like imbibants. Okay, then imbibate. Okay, imbibant means what here? The solid substances. Okay, the solid substances which take part in imbibition. The solid substances which take part in imbibition are called as imbibants. Okay, say for example seeds, then dry wood, etc. Okay, and the liquid part. Okay, usually water. The liquid part or usually water which is imbibed, is known as imbibate. Okay, imbibate. Imbibant is different, imbibate is different, right? And here imbibants are nothing but the seeds and wood. Imbibate means what here? It is the liquid portion, normally water, right? Okay, and these are the two important conditions for the imbibition process. What are those? Difference in concentration gradient, like water potential gradient between the absorbent. Okay, absorbent is also nothing but what here? You can consider it as the imbibant only, right? Between the absorbent or the imbibant and the liquid imbibed. Liquid is considered as the imbibate, right? And affinity between the absorbent and the liquid, right? And here one more example we can give here. Okay, when you soak some uh, raisins, okay, or dry grapes in water during night time, when you observe them early morning, they show swelling. It is due to imbibition only. And when you soak some dried seeds in water in night time, and when you observe in the early morning, again they also show the swelling. It is due to the imbibition only, right? And during this imbibition pressure, what will be developed here? Imbibition. What will be developed? Imbibitional pressure will be developed, right? Okay. And this is the brief account about the imbibition process, right? Okay. And in the next class we will continue with the one more concept called long distance transport of water. Long distance transport of water. Okay. uh, Thank you.